早安，谢谢你邀请. And that's about as much as I'm going to say in Chinese. So thank you very much for the invitation. So I want to really walk you through in this first talk as a physician scientist how we use antimicrobial resistance as a major public health threat and how that actually impacts human health. And what are possible strategies that we can use to counteract this phenomenon? I don't have any conflicts. So this is the only conflict I have. And since I don't know you and you don't know me, I wanted to first tell you where I come from. This is the Houston, Texas Medical Center. Like everything in Texas, everything is big. This is the largest medical center in the world. We have more than 9,000 beds in basically 4 blocks. It's very active with more than 10 million encounters per year. We have the largest cancer center in the country and the largest pediatric hospital in the US. So this is a lot of really interesting geographic concentration of medical institutions with very collaborative environment. My hospital is called Houston Methodist Hospital, which is affiliated with Weill Cornell Medical College in New York, with a research and academic institute, with affiliation with other Texas medical schools as well. And we are a very active hospital in terms of transplant and immunocompromised patients. Just last year, in May 4, 2023, we completed 10,000 organ transplants, mostly liver, with liver and multi-organ transplants. So we have a big population of immunocompromised patients in which antibiotic resistance is really a problem. Okay, I'm going to use this phrase to link the entire talk. And I'm just going to say this, that antibiotic resistance is a natural evolutionary step for microorganisms, and therefore is unavoidable. Okay? So resistance will emerge no matter what you do. So the whole thing on this is how you mitigate that problem. With that, I want to define the current burden of antimicrobial resistance and the future implications for global health. Review as a physician as I am, what is the impact in patients? Understand specific patterns of dissemination of these organisms, some host pathogen interaction at the microbiome level that actually influences development of resistance, and elaborate on some mechanism with a specific example and how we think about this differently. Just yesterday, the United Nations had this big meeting, which there was a political declaration as antimicrobial resistance. The projection with this paper that was published last year indicates that about 39 million people can die of antibiotic-resistant infections by 2050. In 2021, about 1.1 million people died with infections attributable to antimicrobial resistance. Interesting in this report, thanks to efforts in sanitation, the mortality in children have decreased, but has increased in the older population. And all the developed countries and developing countries are really getting older, and these patients will become vulnerable to real antimicrobial resistant organisms. Methicillin-resistant Staph aureus and carbapenem-resistant in gram-negative are the 2 major problems, public health problems, according to this report. So here, I want to show you that the top organisms, Staph aureus, the Enterobacterales, Strep pneumoniae, and others, the major 2 syndromes that cause problems are respiratory infection, lower respiratory infections, bloodstream infection, and eventually intra-abdominal infection. So those are the top 3 problems that these organisms actually cause. And if you think about the number of deaths, more than 120,000 deaths per year goes, for example, for MRSA, and a little bit lower with carbapenem-resistant Enterobacterales. So if you think about what we had in COVID and other infectious diseases, these are big, big numbers, and really surpasses many of the infectious disease threats. So this is a top public health problem, and I hope that the UN declaration help us map a road with urgency to change things in the future. One of the sobering messages of this report is that in the forecasting of antimicrobial resistant deaths, in here, put in red where this is going to be worse in 2050, the region of Latin America where I'm from, I'm Colombian, Central America and Southeast Asia, just in the border with China, are going to be the regions that are going to be more affected by this problem. So this is a global problem that is going to affect us all for these situations. Okay? So with that perspective, let's go back to what it means for a clinician treating a patient. And I brought this paper that was published in 1899. Remember, 1899, we have no antibiotics. This is a case of a heart infection called endocarditis by a microorganism that was called at that time Micrococcus cymogenes, and is now called Enterococcus fecalis. So the infection got to the heart of the patient, and this is a beautiful description of the pathology, physiology, and eventually the patient died because there's no antibiotics, no way to treat this infection. So they ended up dying. They developed interesting animal models and all this really beautiful pathological description. This is something that I recommend to all my medical students when they start medicine to understand how this is done. Let's move more than 100 years later. This is Dr. Munita now in Chile, who was a fellow at one of the cancer centers in Houston. This is a patient that had a bloodstream infection with a similar organism, in this case Enterococcus faecium, who in 2014 or 2012, this patient received all the latest antibiotics for this infection. We could not clear the bloodstream of this patient. The patient ended up dying with blood cultures positive the day the patient died. Most importantly, the two drugs that are important for use against these organisms, daptomycin and lasolid, come from a low MIC to become highly resistant. So this is like an in vivo experiment in a human being that failed. So more than 100 years of that patient in 1899, now in the 21st century, we are running out of options. And this is the main problem that we are facing, particularly in hospitals like mine, where we have really immunocompromised patients affected with these infections. So let's look at this in a broad sense in regions. We've done a lot of work in Latin America, for example, where it is, I mentioned to you, this is one of the focus of this epidemic. And let's look at, for example, at the Staph aureus. We've been looking at the Staph aureus epidemiology for several years now. And you can see this is a study that we did in 10 countries of Latin America, 3 hospital isolates from bacteremia. And what the pies mean are the genetic lineages after whole genome sequences of this. You see in neighboring countries like Colombia, Ecuador, and in this Peru and Chile, you see that the pies have different color. For example, this green color means this is a specific clone called the Cordobese Chilean MRSA, that is ST5. But if you go to Colombia, for example, there is a different clone circulating in this patient. This is actually a USA 300 type ST8 MRSA that I'm going to talk a little bit about you. And this is the people who have done the work there, collaborators, Ginette Reyes, Lorena, and Rafael Rios. So the message here is that this epidemiology is local. Is geographical. You need to understand what is circulating in your own locale to understand the epidemiology. And this is a story, for example, that they do. In the late 1990s, early 2000s, in the United States, there was this epidemic of a community-associated MRSA with severe infections, soft tissue infections and pneumonia, for example, and very confined to the United States. Interestingly, in Colombia, was the only country that a similar epidemic happened. And we had the fortune to follow this in different cities in Colombia. At that time, we used pulse-field gel electrophoresis to type those organisms, and we have like ST types. Interestingly, Colombia was the only country that had a very similar epidemic to the United States, but these organisms, at least with this limited interaction that were ST8, had differences. One of the main differences is that the DNA that carries this genetic determinant MECA was definitely different. So we had the opportunity with Paul Planet and George Weinstock, rest in peace, that we had all this collection of isolates and understand the genomic epidemiology of these organisms. And what we found is that these epidemics had a common ancestor. And something happened that basically divided these epidemics. In blue is the North American epidemic of MRSA, they called the USA 300, and then the South American epidemic in red. So we dig a bit deeper to understand, using this molecular clone and all the genomes, we could identify that around the 1970s, depicted here, there was this discrepancy in this branch, is where they actually separated. In blue, the North American epidemic, and in red, the South American, Latin American variant. And when you look at the genomes very clearly, there was two genetic elements, that one is called ACME, and the other we call COMER. And I'll explain to you in a minute. So ACME stands for arginine catabolic metabolic element that has been shown that seems to help Staph aureus to survive in the skin, and that was characterized in North America in several of these strains. But in Latin America, that piece of DNA was replaced by this island called COMER that carries copper and mercury resistant genes. So heavy metal resistance was rampant in this, and lo and behold, for example, the water in Bogota has the highest concentration of mercury almost in the continent. So this is very well adapted strain, probably driven by contamination with heavy metal in the environment, linking specifically the environment and the changes we are seeing with antibiotic resistance. Chile is experiencing a similar situation, and Chile is a mining country with high-level contamination of heavy metals. And we have identified similar strains in Chile that now are acquiring other resistant determinants and then spreading throughout the country. So this is the most clear link of how we're doing to the planet and how it influences this in human health. So what are the clinical implications? Let's go back to now Klebsiella pneumoniae. This is one of the largest studies that was performed in different regions in the world, including here in China. I was able to participate. This is Antimicrobial Resistant Leadership Group led. And just focus in Klebsiella pneumoniae. So with the ability of clinical data of patients of these different regions, we could see the different clinical effect of these infections in 3 regions in the world: China, South America, and the United States. As you can see, this is what we call a desirability of outcome ranking, which you can see different outcomes based on if the patient is alive, let's say, recover, or with several events, like, for example, renal failure, or the patient's in the ICU. The patient is alive, but not in a good condition. And the another extreme, death. As you can see, in South America, particularly this purple box, is where more people died. And this is correlates with the increased amount of carbapenemase resistant genes that these organisms carry in South America, particularly in Colombia and other countries. This is China, for example, where this is the main box that gets about 23%, but that was very comparable, the mortality with the United States, with no statistical significance. So not only there are different organisms that circulate, but the in-clinical impact depends on many factors and have different outcomes in each world region. Okay. So with that understanding, let's move on to one important factor, which is the same theme. If you give an antibiotic, you're going to develop resistance. And we humans carry bugs all over our place, our body, especially in our microbiome. And there is very clear correlation this now with how resistance emerges and what is the affection of the microbiome. This is work coming from Eric Paymer that was at Sloan Kettering at that time. So when you give an antibiotic, that usually gets into the gut, you disrupt this healthy microbiome that not only is healthy for interactions, but also stimulates the immune system to produce certain antimicrobial peptides that, for example, kill enterococci and maintain these multidrug-resistant bugs at bay, so in low numbers. When you give antibiotics, particularly with anaerobic spectrum, you kill all the good gram-negative organisms. But you not only helps the niche for enterococci to blossom, but also decreases the stimulation of the immune system to produce these antimicrobial peptides. So this is the perfect storm for example, a multidrug-resistant organism to totally dominate the microbiome of patients, particularly hospitalized patients. And with that, you get infections, repeated infections, so you cannot treat for chemotherapy, you cannot give specific therapies, and these patients are really at risk. So I'm going to show you an example. That's something that we are doing in our hospital. This is a project called Dynamite that investigates the dynamics of colonization of multidrug-resistant organisms in the gut, ESBL, CRE, and VRE in ICU patients. These are the first 200 patients. Each dot represents a sample, either a blood, oral swab, or stool sample, and we do a lot of omics, a lot of things with this. I don't have time to show you the results. I just want to give you the clinical impact of this. So if you are in the ICU and you are colonized by any of these organisms, your risk of death increases significantly. If you are colonized with ESBL or CRE, it doesn't seem to be a problem if you separate, at least not correlating. But if you are with VRE, these organisms that typically colonize the gut, your risk of death increases also significantly. If you are consecutively samples yielding these organisms colonized, your risk also increases. If they are very separated, they may not. So that means there is some recovery of your gut. So this complex process of colonization and infection in very critically ill patients is clear to understand how you deploy therapies in this patient, and that's why these patients eventually die. Okay. The other important thing is that let's look now from the patient, let's look to the bug, to the organism. So Shelby Simard, now a doctor, she just graduated, and her project is to understand what happens with the organisms when they are not responding to the antibiotics. This is what we call recalcitrant. I'm going to use vancomycin-resistant enterococci also as an example. So this is another cohort that we call Venus for more than 1,000 patients. And she identified about 41 patients. This is a global cohort, United States, South America, and Europe, that actually we could not clear the bloodstream from these organisms. Either we could not clear what we call persistent or recurring, that potentially the organism came back. So she analyzed using really different genomic tools to do this. And this is a graph that shows the single nucleotide polymorphisms of these organisms from all these patients. That start with here, and then the number of SNPs changes that is graphed here. 20 is sort of the cutoff when you say is the same strain. As you can see here, the majority of dots correspond to the same strain. So there are mutations. So this is how the bugs evolved. A couple of instances, there were major mutations that they say this is a different strain, although it's probably a mutation. So this is how the organisms adapt under the presence of antibiotic and the immune system at the same time. So she interrogated, so what are these genes that change specifically? In those species, fission in fecalis. In fecalis, when you compare to the non-persistent, this is different functional classes. As you can see, this blue dot is recombination and different genes involving recombination. Although it was not statistically significant, these recombination genes were the most common changes in Enterococcus fission. And when you see there was no major difference or statistically significant with the not persistent, but when you do long-read sequencing, you can see these structural variations in the genome. So E-fission evolves based on recombination and changing the genome, but the sequences remain the same. In Enterococcus faecalis, for example, we found two significant group of genes, one involved in carbohydrate metabolism, and the other were actually unknown genes. So the evolution in faecalis seems to be more specific about metabolic needs than was in fission. So this tells you how complex this evolution in the human being in a clinical setting is when they develop resistance. Okay? So then we are going to do now to a more specific. So let's look at how the bug becomes resistant and how they do it. We have been like more than 10 years studying the mechanism of resistance to this antibiotic called daptomycin, which is considered now the frontline antibiotic against Enterococcus and many gram-positives, including staph. This is a lipopeptide antibiotic that has calcium, requires calcium, so gets positively charged, and is very similar to the molecules that the innate produces, like antimicrobial peptides like LL37. So this has been an absolute interesting journey to understand how gram-positive bacteria modify the cell membranes or the cell envelopes to really become resistant. Because as you can imagine, every infection they actually do and kill and encounter this. So daptomycin actually concentrates in the septum. This is Bacillus subtilis, this is the septum, this is fluorescent daptomycin. As you can see, targets the septum where cell division and peptidoglycan synthesis occur. And our latest data and several other groups suggest that daptomycin actually inhibits peptidoglycan synthesis, even though it interacts with the cell membrane due to a lipophilic tail on this. Daptomycin, depicted here in red, interacts with the C55 in areas rich of phospholipids, particularly phosphatidylglycerol, which is a negatively charged phospholipid, so there is interaction that go to the membrane, and then stops lipid 2 interaction. The interaction with daptomycin with the membrane also displaces certain protein like MURG, important for peptidoglycan synthesis. So there are major cell envelope changes that occur within the attack of daptomycin and therefore antimicrobial peptides. So but in the clinic, resistance is very frequent. So how that happens? So we know that there are two main mechanisms in which bugs do this. One, what we call repulsion, mostly effusion and staph aureus, where they change the charge surface of the cell, and positive versus positive, I told you about daptomycin, there is repulsion. So we call the repulsion mechanism. But Enterococcus faecalis, instead of doing this, they do what we call divert the antibiotic from the septum, and I told you they focus on the septum, and they trap this daptomycin in these lipid rafts that are negatively charged and making the antibiotic somehow inefficient to kill. So again, going back to a patient, this is a strain that is from a patient before the daptomycin, a susceptible strain, and this after therapy with daptomycin. We are staining this with this stain called NAO stain, nonil acrid orange. So Cecilia Tran is a PharmD that works on this. And this actually concentrates in the phospholipids, anionic phospholipids of the cell membrane. As you can see, it's bright here in the septum where they should be, and sometimes when the cell divides in the poles. The resistant organisms has a complete different pattern. You see a staining, but not on the septum. So whole genome sequencing and other situation, we found that there was this mutation in this 3-component regulatory system called LIA-FSR. For LIA is lipid 2 interfering antibiotics. This is a response regulator and a histone kinase and a protein that we know now is activator. And this simple mutation, a change of a hylosolution in position 177, was sufficient when we introduced this allele in the susceptible to completely change the cell membrane and become resistant to daptomycin. It's one single mutation completely alters the cell envelope of these organisms. In many studies, we identified these organisms where the target was this gene that encodes a protein called LIA-X of about 533 amino acids. So LIA-X has turned out to be a very interesting. So Aisha Khan, that she was a PhD student at that time, now she is a clinical microbiologist at USC, This is an alpha fold. This is a protein that is predicted an N-terminal domain and a C-terminal with beta-plated sheets. And when you delete either the C-terminal domain or the entire protein, you have the same effect. You see here the now staining in the septum, and then you see the different patterns, and you can see this with a specific microscopy how the patterns change. When you complement the gene, then the now stain going to the septum. So this is the actual effect on this, and we know this protein sits in the surface with the C-terminal disc in the membrane, and probably the N-terminal released to the environment. Most importantly, when the bugs become resistant to daptomycin, the susceptibility to beta-lactams changes dramatically. So this is what we call CISO effect, and that's very important. I'll tell you a little bit about that in a minute. So this is a model of this protein. So this is to confirm this April, another graduate student, and Cecilia, this stain the cells now with this called HADA. I hope you see this. So this is the amino acid, D-alanine, stained with a specific compound that turns out blue. So you can contrast the now staining, and you can see clearly in the resistant strains that are mutants how the concentration of the phospholipids are away from the septum, and you can easily measure it there. And on top of that, this N-terminal seems to bind daptomycin and then also bind antimicrobial peptides like L37. So this is a novel protein that sits in the membrane, probably very close to the membrane, although it doesn't have transmembrane domains, that controls the whole cell envelope, but also binds these drugs. And after several studies, we've shown that this acts as a sentinel molecule that binds antibiotics, and this is like a second messenger for the cell to trigger and maintain this response. So when the system is off, this protein sits in the surface of the cell and is not activated. When the cell is attacked by either daptomycin or antimicrobial peptides, the system is increased, the N-terminal is released, binds daptomycin, and that serves as a sentinel molecule and a signal transduction mechanism to maintain the cell envelope there. Okay? So it's pretty cool, I think, what they do with one single protein. But as a clinician, so what does it tell me? Can we use this to do something? So the first thing that we thought is that these mechanisms of the CISO effect, as you can see, these are two organisms that are daptomycin resistant. These are the normal beta-lactam susceptibilities. First, there is this biological cause with these organisms become susceptible to beta-lactams. So we wanted to know why. So the first step was with Aisha is to stain these organisms and the mutants, and to the laboratory shown here, and two clinical susceptible or resistant. You see these are the resistant strain in red, and this is the MIC for ceftriaxone. And you see a very similar pattern in the penicillin binding protein. So bacillin binds to the penicillin binding proteins. In the susceptible, they bind at the septums, but in the resistant strains, just disperse around the cell. This is FM464 with the stains lipid rafts, and you see the overlay, how they overlap. So the PVP also seem to move away from the septum. So what does it mean? So using the same bacillin, Aisha found that one of the consequences is you separate the PVPs. There are several PVPs, but you concentrate in this class A and class B PVP called PON-A and PVP5, and the intensity of these bands increases in the resistant strains shown red, and this is quantified here. So the movement, the alteration of localization of this PVP increases the affinity for the beta-lactams, and therefore this produces increased susceptibility for beta-lactam, which is very useful for me to treat an organism that is untreatable. So this is the biological cause of resistance. Moreover, we have developed, one of the problems that we have in clinic is daptomycin susceptibility really sucks. It's not really accurate in the clinical strain. This is a work we did with Rodney Humphreys, in which we sent different strains to very prestigious laboratories in the United States. And you can see each bar is one laboratory. They will report in these values all over the place because it's difficult to do. So this is Didra as a house with Cecilia developed an ELISA test that is serving as a surrogate marker first to detect resistance very accurately, but within the susceptible strains, we can detect the ones that had increased LIA-X activity and therefore use this to treat the patient or be more aggressive. Finally, we thought that this is such an important drug, what if we can block it? And think about developing a vaccines for this. So Dr. Singh has developed an endocarditis rat model where we can produce endocarditis and then we infect in the tail vein. And we immunize these rats 2 weeks before this is the inoculum, in 2 groups without immunization, in blue immunized rats, and in green, we call sham immunized when we only do the adjuvant. And as you can see here, this is the number of bacteria per vegetation 2 weeks after the immunization and about 3 days after the challenge with bacteria. You can see a significant decrease in the number of bacteria after immunization. So the concept of immunizing with a resistant determinant is really very new, and this shows very promising results to think about how we can use that against multi-drug-resistant organisms. So with that, I want to conclude telling you that antimicrobial resistance is complex, is a phenomenon that is an important public health problem, particularly in developing countries, that if we understand in detail the mechanism, we can develop really important tools to do that, but we need a comprehensive approach that needs basic science, translational and clinical science to be able to really mitigate the effects of resistance. I told you about several of these collaborators. I want to mention Diana Paneso and Blake Hanson, Blake at the University of Texas School of Public Health. I also want to dedicate this talk, so special in this place, to Dr. Barbara Murray, my mentor, who unfortunately passed away, but she was an inspiration and a leader in this field. Think about all the collaborators of all these projects I told you, some of the grant funding. And to finalize, I want to invite you to listen to our podcast in AAC, Editors in Conversation, that is also done with Journal of Clinical Microbiology. This is my Twitter handle, @SuperbugDog. We publish all that, so follow me. The latest episode is about heteroresistance, which is a very interesting phenomenon. With that, I want to thank you, everybody, for your attention, and Che.
Thank you for your great presentation. Any questions from the audience? 在那个那儿有，两边都有。Yeah.
Dear Professor Arias, thanks for your talk. So our lab is also focused on the antimicrobial peptide. So I feel so excited. But our peptide are de novo designed by deep learning, and it has multi-functions both for virus and bacteria. So my question is, what's your perspective on the future field? Future application of AMPs in this field? Yeah. Thank you.
Antimicrobial peptides are very popular.
Yeah.
In fact, in AAC, we receive a significant amount of antimicrobial peptide papers that, in general, they have a very similar mechanism of action, but for many reasons, it's very easy for bacteria to become resistant, right? Yeah. And that's the reason why we only have very few that eventually made it to the clinical. So my perspective is that from those initial screening, you probably need to work out a more detailed mechanism to try to understand first how they become resistant and what will be the novel aspect that this could offer for what is existing out there. And the mechanism, particularly in gram-positives, is related to membrane interaction. So you really need to think about how to exclusively put your peptide in a different realm to be able to really kill bacteria in a very detailed manner.
Okay, thank you very much.
Here.
Thank you for your presentation. I followed your presentation with keen interest, especially when you showed the map of distribution of antimicrobial resistance. I can see China and Africa are safe for now. What do you think is responsible for our safety? I mean, why is it that in Africa, for example, and China, this issue is not a problem for now, so that we can see how we can try to prevent ourselves from that? Thank you.
Yeah, thank you very much for pointing that out. I can tell you that for the data in Africa, you read the paper, the reason there is no stronger color there is because there is a lack of data. Okay? So it's more a lack of data than the reality, the real problem that it is. In China, I think they are becoming much more aware, and we know, we collaborated with some of the researchers here that is being done easily. But the lack of data preclude a good projection to what's happening in Africa. But I think the situation is really dire.
So Cesar, I have a question. So Staph aureus, Enterococcus, these are the bacteria or Klebsiella. They are not naturally competent for transformation, but on the other hand, they are the leading bacteria that carry this resistance genes and exchange those elements across the species or internal within the species. How do you think that mechanistically happening?
So yeah, I probably need an hour to answer that question. But so the transmission thing is something that is interesting. So at least with MRSA, it's clearly that there is contact in patients, and patients, for example, community-associated MRSA share the same strain. So there's definitely dissemination between, mostly for contact. Most of the gene exchange that occurs seem to occur with the microbiota of the organism, and there's a very good example, including the MEC gene in Staph aureus.
Do you think it's through conjugation? You said physical contact means conjugation-driven?
No, I was talking patient physical contact, and in bacteria, in the microbiota, several mechanisms. Pneumococcus, for example, is a master in transformation, so they can pick up gene naked DNA, and that's how they became resistant to penicillin. Enterococci has many, many abilities to carry and capture particularly genes by conjugation, and the lack of CRISPRs, for example, in Enterococcus fishes have been shown how they acquire this gene. Also, their genomes are incredibly malleable, so that's something that has really allowed them to evolve, as I showed you in some of these slides.
Okay, given the time limit, thanks again for Arias' wonderful presentation. Thank you very much.
Cheers.
Now